Heinz body test. Heinz body we have already seen in under G6PD deficiency. What are Heinz bodies, from how Heinz bodies are produced. In this session we will see how these Heinz bodies are tested. Heinz bodies are late signs of oxidative damage and represents an end product of degradation of hemoglobin. That is, they are also referred to as Heinz-Ehrlich bodies, and they appear as small round inclusions within the red cell body. They are late signs of oxidative damage and represents end product of denatured hemoglobin. They are not visible when stained with Romanowski stains. So, a special stain is required, that is supravital staining. Example for supravital staining are new methylene blue, crystal violet or bromocrysyl blue. Associated disorders, that is under which this Heinz bodies are seen. Heinz bodies are seen in case of thalassemia, glucose 6-phosphate dehydrogenase, etc. Principle of the test. The test depends on abnormal sensitivity of G6PD deficient red cells to the toxic effect of phenylhydrazine. The drug causes the hemoglobin to be denatured and precipitated as small round inclusions which can be seen on staining. The reagents required here are heparinized blood, that is blood collected in heparin anticoagulant, buffered saline, that is Sorensen's buffer is required. Solution A, disodium hydrogen phosphate 0.15 molar, dissolve 11.876 gram per liter of distilled water. Solution B, potassium dihydrogen phosphate 0.15 molar, dissolve 9.078 gram per liter of distilled water. Mix 10 ml of solution B, that is reagent 2, with 90 ml of reagent 1 with a final pH of 7.6. The third reagent required or requirement is 0.1% weight by volume acetyl phenylhydrazine solution. Preparation: acetyl phenylhydrazine 10 milligrams, Sorensen's phosphate buffer 5 ml, normal saline 5 ml. So this gives 0.1% acetyl phenylhydrazine solution. Next reagent required is 0.5% methyl violet stain. Methyl violet stain 0.5 gram, normal saline 100 ml. Dissolve dye in saline and filter it. Do not do the test within 1 hour of drawing the blood. The reason is you won't get a clear picture of Heinz bodies. Now the procedure. Collect 2 ml of venous blood from patient and also from the normal person in an in an heparin anticoagulated tube. Mix the samples well. Set up 2 test tubes and label it as patient or test and control. Add 2 ml of acetyl phenylhydrazine into each of the test tubes. Add to the test tube, add 0.1 ml of patient's blood, and to the control sample tube add 0.1 ml of the control sample blood, heparinized blood. Mix and aerate by blowing through the pipette a few times. That is called as aeration. You provide air. Incubate tubes in 37 degree centigrade water bath for about 2 hours. Repeat aeration and continue incubation for another 2 hours. Mix. Place 4 drops of methyl violet solution on a clean microscope slide. Add 1 drop of the blood and mix it with it, or what preparation you have done, from that 1 drop and mix it. Place a cover slip and ring the edges with Vaseline or any other sealing agents also. You can use paraffin wax, nail polish, etc. to seal. Stand at room temperature for about 10 minutes. Observe under microscope and a high power objective with reduced light. Heinz bodies appear purple round inclusions of varying sizes within the red cells. Count 100 RBCs. Any cell containing 5 or more Heinz bodies are counted as positive. Result. First count the control blood sample. If it is less than 30%, then the test condition have been satisfactory, since in case of normal person, less than 30% RBCs will only have 5 or more Heinz bodies when mixed with an acetylphenyl hydrazine. Now count the patient's sample. If the result is less than 30%, the patient is normal. If the result is more than 40%, the result is abnormal or positive Heinz body test. Interpretation. Abnormal results or positive Heinz body test are found in people with complete absence or considerably defect in the red cell G6PD enzyme. Second, patients whose blood has been exposed to various drugs like sulfur group containing drugs or inorganic oxidizing agents such as potassium chlorate. Third, people with unstable hemoglobins. This is about Heinz body test. Next we move on to the preparation of hemolysate. What is hemolysate? Why the preparation of hemolysate is required? Principle will see. When tests have to be done on hemoglobin, we have to first release the hemoglobin from within the RBCs. That is lysis. The liquid is released, which is called as hemolysate, and contain free hemoglobin in it. The RBCs are washed before the hemolysate is prepared so that no other plasma proteins or lipid content is present. Next, moving on to the method. Collect 8 to 10 ml of blood, that is EDTA anticoagulated blood, into a tube. Centrifuge at 2500 rpm for about 5 minutes. Aspirate and discard the supernatant plasma. We do not require plasma here, we want only the red cells. Add 3/4 of the test tube with the saline, normal saline, that is 9 gram per dl of saline, 9 gram per liter of saline, and mix the saline and red cells completely. This procedure is called as washing. Little bit of red cells in the test tube. To that you add 3/4 of the test tube with saline and mix the content. Now centrifuge at 2500 rpm for about 5 minutes. Then aspirate and discard the supernatant. We do not require the supernatant. Now this procedure is washing. Repeat this procedure for about 2 more times. And the last wash what you do, the supernatant is discarded. We do not want the supernatant. The sediment what you get is the packed red cells. These red cells are then lysed by using different methods. So we will see what are the different methods used for lysing the packed red cells. So the remaining washed packed red cells are lysed using by any one of the following methods. So the first method is add double the volume of distilled water and 1 volume of carbon tetrachloride. That is, suppose you have 1 ml of the packed red cells, then you will add 2 ml of distilled water and 1 ml of carbon tetrachloride solution. So that will give you a hemolysate solution. Second method is adding few milligrams of saponin. The saponin chemicals also help to lyse the red cell. The third method is freezing and thawing of the red cells at room temperature and then add 2 volume of carbon tetrachloride. Freezing is you freeze the red cells. Once packed red cells are got, they are frozen in the freezer. Then take out the red cells, frozen red cells, and keep it in the room temperature. The frozen red cells, when it returns to room temperature, the procedure is called as thawing. Then to this thawed red cells you add 2 volume, that is if 1 ml of thawed red cell is there, to that you add 2 ml of carbon tetrachloride solution. These are the different methods of preparing lysis of red cell. Now after lysing, the cell in the tube are mixed in a mechanical agitator or shaken by hand. Centrifuge the content at 3000 rpm for about 30 minutes. Transfer the supernatant to a clean tube. Check the hemoglobin level. Adjust the hemoglobin level to about 10 to 15 gram%. For adjusting, you can use distilled water. Do not dilute the hemoglobin level below 10 gram%. Uses of this hemolyzate. The hemolyzate can be kept for some weeks, and it is used for hemoglobin electrophoresis, fetal hemoglobin estimation, hemoglobin A2 estimation, that is adult 2 hemoglobin estimation, and unstable hemoglobin tests.